guess we can start. Let's start directly.
Hi. Hello, everyone. Sorry about the late. I was just here 5 minutes ago to look up my slides. Thanks, Hernando, for the invitation and putting me up as the first speaker with a loaded section titled called Causality. I'll try to connect to what I've been doing. It's based on graphical modeling in Fourier domain for large dimensional time series. It's joint work with Nobonil and Yohun, who is a grad student and postdoc in Cornell, Amy Kujaski, who's a computational neuroscientist in Cornell, and Suhasini Subbarao, who's a statistician in Texas A&M. So 15 minutes, I won't be able to get through all of it. So this is the main takeaways. In my research experience so far, using Gaussian graphical models, networks, or many other fancy tools, we can only search for a potential cause, but not really establish causality without randomized experiments. And that's the limitation I guess we need to keep in mind. But that being said, I would say the path we take, the questions we ask, the methods we design to do this search is extremely helpful. Even if it does not help us establish causality, I would say it definitely helps us narrow down a potential set of causes. And from there, we could do more deep dive using domain knowledge, of course, what causes what. So a crucial step that Gaussian graphical models help us do is partial out effects of many observed confounders. And when we work with large-scale signals or time series, as in brain signals from different brain regions, a promising direction seems to be a marriage of spectral analysis, Fourier domain analysis with high-dimensional statistics. So doing Gaussian graphical model precision matrix estimation type work, but on the Fourier transform of the data. So two obvious challenges we faced in our group while trying to do this. First is, if you're working with complex variate data and you want to do high dimensional statistics, how do you design optimization algorithm? What can you tell about their guarantees? How do you know they're fast? It's somewhat away from what I've been trained with. I have taken a course on complex analysis, but I feel bad to tell my graduate student, go read all about holomorphic functions before doing time series modeling. So we thought we have to come up with some middle ground where we can comfortably think about this complex variable graphical model with already what we know in high dimensional stats. And the second part is statistical inference with complex Gaussians in high dimension. That remains challenging. We are making some progress, but it's interesting thing to think about. How do we even think about variance of a complex number? There's a real number, there is an imaginary component, there are two variances, a covariance together. When you go high dimension, how do you reduce this dimensionality? What do you focus on? So there are interesting statistical questions that can be asked. So some progress on my side. To answer the optimization question, we were able to come up with a realification strategy. So when we have complex numbers, complex scalars, vector matrices, we can, like liquefaction, we can realify them using some tools called ring isomorphism in abstract algebra, and that leads to really fast algorithms. So I'll share some results. We show that in high dimension, we can do consistent estimations. So statistically with penalty and everything, we still have benefits. In the next step, we are trying to do adaptive bandwidth selection, which seems to me like one big open area in this process. In asymptotics, we don't talk much about the right bandwidth selection. We go with bandwidth need to be selected high or in the right order, but in practice, it makes a huge difference. And as I said, understanding more about variance of complex random variables. So this is some directions where I think statisticians' expertise could be helpful. So I'll go at very high level for the rest of this time, but all the technical details can be found in these two papers. First one we submitted recently, second one appeared in Annals last year, I think. All right, so I did graphical model in economics and finance quite a bit. So we would study the evolution of trajectories of stock returns of different companies and try to build this type of directed graphical models. So usually it goes by the name of Granger causality, which is some form of a predictive pattern. What it essentially looks for is lead lag pattern. For example, here you see Bank of America stock return dipped, a little bit later Goldman Sachs did. So in my graph, I am tempted to put an arrow here, Bank of America to Goldman Sachs, and that's like the major criticism against Granger causality, that you don't account for confounders. If you look at maybe AIG and see AIG dipped earlier and that caused all of this, the true graph should look something like this. So you need to adjust for AIG's past values before drawing this kind of edge, and that's typically done through a formal vector autoregressive model of one order, where you regress the Goldman Sachs return on past return of itself, past return of Bank of America, AIG, all. And when you imagine throwing in many, many confounders in the mix, you throw in your favorite penalty and see if the coefficient of interest is non-zero. So when we try to play the same trick on physiological signals like EEG, we often find very large bar orders. There is so much dependency. I need to throw a lot amount of data and downsample to really get something meaningful, and which was not satisfactory. So we started looking at it through the lens of frequency domain. I tried to summarize the main methodology through this little doodle. So please bear with me and my bad handwriting. So what happens roughly in the Fourier domain graphical model is, imagine you see a brain signal of region 1 and region 2. Region 1, this time series xt, you can break it up as a linear combination of a low frequency component and a high frequency component. And as you can imagine, looking at the relative size of the two, maybe the low frequency has higher weight, the high frequency will have smaller weight. So x1t would be d1, d low frequency 1 times blah, d high frequency 1 times blah. This will be small, this will be large. Compared to region 2, there is a high frequency component has a slightly higher weight. So you can also write xt as a linear combination of these two waves with the corresponding weights. So the essence of frequency domain graphical modeling is presenting each of these series through a sequence of numbers, these coefficients. These are coefficients on the Fourier basis, and coefficients as they should be, they are inner product of the true graph and the basis. And if you want to access these coefficients, you can actually calculate the discrete Fourier transform of your data. So when it comes down to implementing stuff, this is how it looks. I have an n by p matrix, n time point of the x time series of p dimension, p brain regions. I do a fast Fourier transform and get discrete Fourier transform. Now I end up with a n by p complex valued matrix. So this d11 will be around here. dl1 will be around here. So all of these numbers are coded in this complex valued data. So now that I have another matrix, the only difference from what I know in high dimensional stat is it just has complex number, nothing else. I can actually be ambitious and think about looking for correlation, partial correlation, many other things from this data set. And many of the intuitions we have from multivariate high dimensional stats goes through. The challenge is, of course, these are now complex. So I talk about two styles of graphical modeling. First is stationary graphical model. Stationary graphical model asks the question, is this guy and this green guy, are they correlated given the same data from all the other regions? So imagine this region's signal is maybe heartbeat and breathing rate. And what you see happening here is all the components contribution from heartbeat. And you see if the heartbeat component to region 1, region 2 are correlated given everybody else. If not, then you don't put in an edge. If they are, you put in an edge, that kind of graph. Okay? So very simple correlation question between the two. So if I want to do this in my data, what would I do? I zoom in on this section of the data, which is about the frequency of interest. Look at these two columns and look for correlations between them. If I want to do graphical model, you will look at correlation between the two, condition on all the other columns. So this exercise is very much like how we do partial correlation analysis. And it is indeed nothing but calculating partial coherency. But we want to do it in high dimension, so we want to adjust for many, many confounders here. So that line of work is stationary graphical model, and that's in our first paper. And this is where we first looked at all the computational challenges. The second line of work is looking for correlation between frequencies. So what happens here, are they correlated to what happens here or what happens here? Again, condition on all the other regions. Why do I look at correlations between frequencies? So there is a result that if your process is stationary, whatever happens to one frequency, other frequency does not matter. So there should not be any correlation for a stationary process. When you spot correlations, that should be indication of non-stationarity. So by studying these correlations across frequency, again, in data, this would only mean you look up this part of your complex matrix and look for its correlation with this segment or this segment. So somewhat medium frequency to high frequency, what happens to different components. So to formalize these types of associations, we came up with a new notion of conditional non-stationarity. And what it helps is tease out the source of non-stationarity to the effect of non-stationarity. So yeah, these two types of graphical models we built. In the remaining 4 minutes, I quickly talk about some optimization issues that came up in the stationary case. So in the stationary case, our problem was building graphical lasso. So imagine all your discrete Fourier transforms are complex Gaussian valued, and you write down that complex Gaussian likelihood, throw in your favorite penalty, and you want to estimate this. How would you do that, the complex valued graphical lasso? So in prior work, we found Mark's work where they used a Klein-based trick, but separated the real and imaginary part, and we found it artificially increases the dimension. There are other off-the-shelf solvers like ADMM, but they're still not like very scalable in high dimension. In high dimensional stat, the state of the art is pathwise coordinate descent. They look at the sparsity structure of the problem, and there are lots of really clever tricks to make the algorithm scalable by exploiting sparsity. So we wanted to do all of that in the complex domain. And effectively, what it came down to is this very simple observation. How we think about the number I. The imaginary number I, we think of it as a vector. In a two-dimensional plane, I is the vector 0, 1. But that realification did not work. If you think of I as this matrix, because I times 1 is I, so you multiply 1 by I, it goes up by 90 degrees. I times I is minus 1, so you multiply I by I, it also goes up 90 degrees. So you think of I as this 2 by 2 orthogonal matrix and build up. You could show that every complex number can be represented as a 2 by 2 matrix. And this is very special. And in the language of abstract algebra, it gives a field isomorphism, which means if you add, subtract, multiply, divide with the vectors matrix of complexes, the same thing happens on the 2 by 2 real matrices as well. So all the things we know just by making this transformation, we came up with new algorithms called complex valued lasso, which looks like this, very simple, very similar to usual lasso. We had complex valued graphical lasso, and we implemented them in Fortran. And as you can see, they're much, much faster. This is our Fortran implementation, and this is GG lasso, the state-of-the-art group lasso implementation here. And it's also accurate. It's not just fast, it gives you the right result. So as you can see, the regularization path, they are tipping at the right point. So that was one big progress. Now we are looking into more complicated model development in the frequency domain. We applied it on human connectome project fMRI data. We get anticipated sparsification compared to regular partial coherency. Okay, 1.5 minutes. Very high-level idea of what this non-stationary graphical model business is about. Look at this 4 time series. Imagine they're coming from 4 different parts of brain. You can tell they're all non-stationarity. Around here, there is less negative correlation. Here, maybe more positive autocorrelation. Something changed over time. But can you tell where it first began and who affected whom? Probably not. If there is a source, one part of brain where things first became non-stationarity and that hit everybody else, we cannot see this through naked eye. Actually, it's generated from a model where the 1 and 3 are the source, everybody else are the affected guy. So it turns out this type of relation where you have some source and some affected children, you can represent in the form of a new graphical model, which is in our paper. So these are stationary part of the graphical model. These are non-stationary part of the graphical model, the dotted. So with that notation, my 1 and 3 will be the non-stationary part, and these will be stationary condition on the non-stationary part. They are the source. They just get non-stationarity from everybody else, from these guys. And that little doodle picture I showed you before with the pink diagrams doing correlation across frequency, we showed that you can actually pick those up. In this graph, you have 1 and 3 are non-stationarity in this graph. And this other edge, 2, 4, and the red graphs, they only show up here. So just looking at the discrete Fourier transform carefully and doing this high dimensional object, you can build innovative graphical model that tells you more about your data. So yeah, I'll stop here.
Because of time, we will directly move on to the second talk. So, so our second speaker is Professor Rossi from UTHealth Houston. So let's welcome.
Um, Thanks home to Jen and Hernando and also the committee for the invitation. And I really enjoyed this workshop. And all because of the loaded title about causality, I probably cannot say too much into on that big topic. So I will just share some of those experience when in the causal mediation in those type of models from fMRI. So the fMRI experiments we can mainly focus on will be those task fMRI. And even though the stimuli are randomized and the data we can analyze are not totally randomized. So there are some issues when we're trying to apply causality to the data in fMRI. And this is different from resting state. And I think when I started looking at this, because my graduate training is mainly in sort of like a machine learning, data-driven type of research, and then I come across this and saying you cannot get causality from data. Well, that's not so encouraging when you're just trying to look at the area. So, but we'll give it some try, and so some of the work we are really trying to relax those assumptions and so-called untestable or untested assumptions. And one of the challenge that we face, fMRI, as many of the speakers have in this workshop has mentioned, is that we have really multi-level data. And nowadays, with large cohort data, population studies, we can easily go up to millions of data points or even more if you count each individual voxels cross time and cross subjects and also cross sessions. And later, we'll come back to this. Actually, multi-level can help us a bit trying to resolve those causality issues. And one of the model that I really want to build on is to think about, and when you have the stimulus goes into the brain, how the stimulus information is really processed in the brain. And we are really taking a baby step. And ideally, we want to find out all the wonderful voxels at the levels like many of the speakers mentioned about on those microns or those fibers. But today we just focus on, let's say, how do we even understand you have two brain regions, how the information is processed across those two regions, whether it goes through the blue path or whether it goes through the red path. And so in the mediation literature, it can be called indirect effects and direct effects. And one of the first attempt to trying to think about this is in the causal literature, there is always so-called unmeasured confounding, which is a big issue trying to resolve the causality between the mediator M and R here. And if we simplify the model enough, so those modeling assumptions and Gaussian assumptions and etc., so it can be reduced to whether trying to understand the errors added to those equations to M and R are correlated a lot. So, and that's one of the untestable assumptions, whether they say the delta is going to equal to 0, that means no major confounders. And of course, we are learning a lot from the a growing literature on mediation and reading a lot of them. So I can only list a couple of them. And of course, one of the issues people realize is that you can have totally different causal models that will generate exactly the same data. So that's the really the challenge. So if you have unmeasured confounding. So what we first attempt to do is to leverage the multi-level structure, and for the sake of time, I won't go to the technical details. And because the multi-level structure, actually, if we leveraging those like SPM, those like mixed effect type models and building on those, and if we believe those models are true or reasonably approximate the data. So we put those together with mediation models, and we can estimate the causal effect without only one untestable assumption of the delta. If you remember on the previous slide, we are no longer assuming the delta to be 0. So it means that there is unmeasured confounding between the mediator and the outcome. And so that's one of the lesson we try to learn, is that it seems like we can leverage the large fMRI data, even though it's a challenge for computation and for modeling, but it can help us resolve causality a bit if we sort of leverage that kind of data structure. And as a result, and this is an example that we can show on the left, that we can identify the maximum likelihood as this kind of curve. And because at first that if we don't leverage the multi-level data, the likelihood is actually flat. You cannot identify the delta. So here we do have a unique delta, and which is non-zero, as well as we can look at the estimates, and it's sort of doubled compared with if you just do association analysis or do regression, and you will get that assuming the delta is 0, the effects actually get doubled. So the causal, when you are doing the causal analysis, it actually helps us double the effects, or after removing the measure of confounding. And we also trying to extend that kind of work to time series data, and building on the work of granular causality and the vector autoregressive models. And thanks to the previous speaker has already introduced that. So for here, granular causality alone, of course, is not truly causality. So we also added some assumptions, and in order to really do causal, and we did some theoretical work on the convergence as well. So what ends up with is, if you take a mediation model, which can already consider 3 variables, if you consider that it's on the spatial domain, and then if you consider the vector autoregressive models on the time domain, then you have a spatial temporal process, and it has a lot of arrows going back and forth. So I will not get into the details. And actually, a lot of work is done by thanks to Yi, and figuring out this kind of very complex directions and causalities. And so what we finally see is that we compare with the various other approaches, and it seems like it does match the theory. So usually we see is that when you assuming the delta is 0, and it will be the same as our methods, but you move away from delta when you have larger and larger confounding in terms of magnitude, and you'll see large bias by other methods. And if you don't take into account the confounding issues, and we also have estimates for the so-called AR model parameters here as well. They seem to match the theoretical results. And what we also learned from this work is that when we apply to the fMRI data, we can see is that we can actually find stronger indirect effects and from the data. Well, if other methods, they don't consider those causal estimates, they tend to actually underestimate the effects or the ratios. And one of the measures people use in mediation is really to look at the ratio of the AB and the VS, the sum of AB plus C. And those arrows, as you can see, some of the methods, like the one in purple, they really have small estimates for AB, and for C is very large. So if using our approach, that will be able to reduce the effects for C, and also have larger estimates for AB, and also statistically significant as well. And so this is also driven by some of my long time collaborations with neuroscientists, and we are trying to figure out in risk-taking in the stop-go trials and how the information is processed by the SMA region. And so some other neuroscientists also suggest other models, and I will not get into the details because I'm not a neuroscientist. People talk about top-down or bottom-up and or different pathways. So it seems like the pathway we have published before, like the brain SMA, and seems to be an important one. And we also have found some feedbacks, because for neuroscientists, when we have this kind of time series model, we can also talk about not just the information flows in one directional, but they can also have feedbacks as well. And then we try to attempt this kind of work recently, and to using the functional data idea, and using the functional data, it really gives us a good dynamic and also presentations and also interpretations of the mediation effects. And mathematically, they also have nice graphs and showing direct and indirect effects. And what we learn from this is that when we have those functional data, now all those mediation effects, they look like curves and also the functions. And then we also have to do integrations in order to figure out the effects. And while we feel like really fortunate is that when we consider those functional models, I think here the numbers are really look really nice for us. It's just because that we set up our model to be a functional model, and if we apply a scalar type of mediation models to functional data, it really has functional models really have a big improvement compared with the scalar versions. And so there are some interpretations we can get from applying to real data and look at how the stimulus drives the indirect and direct effects, and depending on the stimulus, its history, and etc. And lastly, I'll just briefly mention that we also thought about trying to extend this to really high dimensional mediation, and we have many mediators, how to figure out those pathways. I think Eric also mentioned about regularization. So we also have did some work in talking about this kind of pathway type penalty, and which helps trying to figure out, as a baby step, trying to figure out is not just thinking about 2 brain regions. What if 3, 4, 5, or even more brain regions? How the informations are getting processed, and hopefully as a summary, that I think the causal mediation analysis for fMRI, sometimes it can be helpful and for the effects, and also it is kind of fortunate, and that we have really large data and multi-level data. Those kind of data structures can help resolve the causality a bit. But of course, we have to make some other assumptions. I think they're pretty reasonable under current understanding of fMRI and experiments, and mediation can also be helpful of hopefully understand the information processing. So even though what we did is really a baby step, we don't fully understand how the brain processes various informations and on a much larger scale. So we started with only 1 or 2 or 3 regions. And so thanks for my wonderful collaborators, and is here. And Martin, we learned a lot on mediation, and Brian and Michael as well. I think Jen and Huntu posted those questions. So important research problem, I think, for causal inference, how to validate causality, that's kind of really challenging for us. We can look at the literature using sparing stimulations or other things, trying to figure out if those identified brain causal pathways are indeed there, and how do we build large-scale type of causal models, and trying to have better understanding of brain pathways and directionality. I think some of the speakers on Tuesday also mentioned that, and how do we to bedside, how do we have link that to those causal models and related to behavior to outcomes, and we don't know. For second, I don't really have too much input. I don't do much. I feel really guilty about not doing that much of processing, and so I'll just say it would be really nice to have a benchmark. And lastly, I think for statistical method, I think we're also good. We just do our packages. I feel like it'd be really nice if we can have those softwares available already in FSL or SPM, or those package people can just click a button. So instead of they have to figure out how to integrate MATLAB with R or try to integrate, and I think I also learned a lot of the points from the speakers at this workshop. So I guess I will try to attend more scientific meetings and more case studies. And well, thank you very much for your attention.
I think we can leave all the questions to the end and directly move on to the third talk.
You're old fashioned, my friend. Why don't you write on the board?
I want to make handwriting great again.
Okay, so our third speaker is Professor Hernando Ombao from KAUST, and here is his title. Let's welcome.
Thank you. Thanks, Ting, for agreeing to
You just
Chair.
use the ChatGPT and dump it inside and it generated the PPT.
yes, but I really like my handwriting. Yeah, thanks, Ting, for chairing the session, and really a big thanks to both Hongtu and Adrian for this wonderful workshop. So today I'd like to give an overview about mixed time series in design animal experiments, and in fact, how causality in its real sense can be better assessed under these settings. So here's a first project that we're working on. This is from a working memory rats data set at the 14 lab at UC Irvine. So here is a rat. A rat is trained to recognize 5 odors, A, B, C, D, E. And there is a sequence of these odors, A, B, C, D, E. And so the rat puts his or her or its nose in that nose poke. And then if it realizes that there is that the sequence was not presented in the proper order, that's off sequence, then the rat is supposed to take the nose out of it, and then the rat gets a reward. So rats are already known to have a very good working memory in space, but here it's a test of how they are able to remember the sequence of odors. So there are 5 odors here. I don't remember which one exactly, but there's popcorn, there's banana, there's cake, these kinds of things. Okay. So the goal of my collaborators, Norbert Fortin and his group, is to study the role of the hippocampus in memory formation and retrieval. So this is an animal study, but it has potential impact for humans. So there's the potential knowledge we can gain from studying animals, in rats in particular, may be helpful in studying dementia, MCI, Alzheimer's disease. And so, yes. Now, in their experiment, they actually implant tetrodes onto the hippocampus, and then they record 2 types of signals. Spike train data, a sequence of 1s and 0s, 1 when there's a spike, 0 when there's not. It's binary time series, and local field potentials. These are continuous valued measurements, just like EEGs, but it's in a very localized field. So here's an example of the data set. At the very top, that's a local field potential recording. The second row is beta power computed at a local time window. The third line is a spike train. And the fourth line, these are rates, again, computed on a small time window. The goal is to study spiking rates in rats, to study the probability of a spike conditional on its own history, the history of other neurons, and the past history of the LFPs, and how this particular spiking pattern may differ whenever a rat is exposed to an in-sequence or an out-of-sequence type of a stimulus. Okay. So here we have LFPs. These are recorded from P different points in space. And we look at features from LFPs. This could be variance or cross correlation, spectrum coherence, and other types of dependence measures. Okay. The second study is by my collaborator, Andrew Holbrook at UCLA, and another collaborator from MGH. This work is funded by the Kavli Foundation and the Karen Toffler Charitable Trust. And in their experiment, they actually have two groups of rats. One is a rat that's the normal type of controls, and the other are the rats who are genetically modified so that they have the tendency to produce abnormally high levels of tau proteins, which are actually biomarkers for dementia. Okay? So you have these two groups, and in this case, one can think of this is quite a nice. One can assess causality in this setting. You have the two groups of rats, one with a quote-unquote treatment, which is actually not a treatment in this case, and then the other is the control. And quite interestingly, for this in this study, it's longitudinal, and so rats have a lifespan of 12 to 24 months, and here they study the rat continuously from age 3 months all the way to 9 months. So in this 6 months lifespan of the study, they expose these rats to some cognitive tasks, and then they study whether there are changes for each rat in terms of their cognition ability over time, and also whether those changes differ between the control rats, group 1, and the rats with genetically modified to produce high levels of tau protein. So in a sense, here is a story that all of us are trying to work with, assist our collaborators. So we have stimuli, interventions, treatments, and so on. And these lead to neuronal activity, which again leads to behavior. When it comes to brain activity, then here we're looking at the ensemble of neurons and also subpopulations of neurons whose activity is measured by local field potentials. So as I said earlier, we want to look at dependency. We want to look at how a neuron at time t, how that behaves, whether or not it spikes as a function of its own past and the past of other neurons, and perhaps also the past of the other LFPs. This is spiking as well, that's one. And you can also look at the number of times that these neurons might, the number of neurons that spike as well, so summing overall K. And I want to compare the spiking tendency between off-sequence and in-sequence, and also between those who are tau protein bred versus the control. So when it comes to dependence among LFP channels, there's not one unique way to characterize dependence. Now, all of these, I would say, are looking at Granger type of causality, Granger type of dependence, unless, of course, there is that intervention with like breeding with the tau protein or otherwise. So these are the different measures of dependence. So I'll go over some of these very in a cartoon way. So thanks to Sumanta, he already drew a similar picture. So if I want to look at dependency between X1 and X2, I can look at correlation between these two channels. But sometimes I want to be able to identify which frequency oscillations drive that dependence. And one way to do that is by decomposing my signal. So you can decompose it into however way you like. But if you're doing Fourier, then you can decompose the first signal into low frequency, and then there's high frequency. There's no middle frequency. Whereas for the other signal, you have low, middle, and no high. And so therefore, if I look at the correlation between these two signals, they will be correlated, and that correlation will be attributed to the presence of low frequencies in these two signals. And in fact, this framework of trying to characterize dependence can be, as I said, generalized to more than just the Fourier. And so one way to think about that is in this framework, you have X1 and X2. X1 and X2 are sums of these oscillations, and the dependency between X1 and X2 can be expressed as the dependency between those, the decomposition, and under certain conditions, the dependency between the sum becomes the sum of the dependencies across each frequency oscillations. And so if I'm looking at covariance between X1 and X2, I can decompose that as a sum of the covariances. And if this value is high, I can then identify the case, the oscillations that contribute the most to that covariance. Now, some of the challenges that we'd like to address here. The first one is more general measures of dependence. So we're thinking about, can we look at the full distribution instead of just the first few moments? We need to take into account nonlinearity and non-stationarity. And this work is being conducted by Paulo Redondo, that of spectral transfer entropy. And so it's looking at how does the knowledge of past oscillatory activity in one channel, in one node, lead to reduced entropy or uncertainty in another channel at a particular oscillation. Okay? So if there is a reduction in entropy and uncertainty, then we conclude that there is Lagrangian causality. Okay? Lagrangian causality is going to be based not only on correlation, but actually in the entire distribution. These are all based on the entire distributions. Now, the other is that of causality. And so here I'm going to appeal that I think we need to offer more support for these animal studies. I think we're all enamored to go into human studies, quite interesting, directly impactful, but I would say that these non-human animal studies are not any less significant than human studies. And in fact, we learn a lot from these animal studies. We are able to manipulate. I mean, for those of you who are animal rights activists, please don't kill me. But with respect to rats, to monkeys, then we're able to do manipulations on them that we cannot do to our fellow human being. And but the studies we get here actually are very, very informative. Now, still open questions. We asked about this a few days ago, is that how do we combine information across different subjects in an experiment with many, many subjects? So there's, even within a subject, there's large variation. Now, very often for biostats, we think about the mixed effects model. So if I have a subject-specific parameter, I want to decompose that into some group or fixed effect, plus the subject's own deviation from that group effect. But this does not always work. And don't insist on it all the time, because sometimes it just doesn't work. It doesn't, you cannot just do that. Sometimes you'll have to content yourself with qualitative descriptions and so on. Now, related to this, related to this is the need to develop distributions of objects like directional networks. I think this was handled earlier today. So people like Sharmista, Lisa Levina are working on similar topics. And so here we want to think about networks as outcomes. That Xavier worked on, discussed today about modeling the correlation matrix as an outcome. Now we also want to be able to compare distributions. So comparing distributions mean that Okay, how do we define the center of that distribution? How do we define the spread? How do we define, quote unquote, the difference, and so on? So all of these are very interesting open questions. How do we measure the strength of evidence? Is it always going to be p-value? I don't know. Maybe likelihood ratio could be one. And similarly here, we want to think about networks as predictors as well. So I mentioned networks as outcomes depending on the group, control, or tau bred, or networks as outcomes with respect to stimulus type, off sequence, in sequence. But networks can also be predictors. Okay? Changes in brain networks can these be used as predictors for dementia or MCI? And here, my second to the last point is that on moving away from pairwise dependence. So we may have to consider the entire network, a more macroscopic, so going from micro to meso to macro. So instead of looking at pairwise dependency between this and so on, we might have to look at subregions, dependencies between these subregions, each of them high dimensional. So again, open problems. We need to look at robust canonical coherence, and perhaps we may consider topological approaches like the ones being developed by Mu Chang. So there's a G here, just did not appear in this PDF. And finally, this is already announced earlier by Marina. So we have now a new journal, Statistics and Data Science and Imaging. This is, Marina is our editor-in-chief, and Michele Guindani, Martin, and I are co-editors. And I'm sorry?
What you need to pay?
Oh,
First year free.
yeah, yes.
Second year, 60.
So submit your best papers.
About 1,500.
1,500. Yeah, 1,600.
Reasonable.
Maybe Marina, if she's here, okay. So we'll have this turned on. I think by Friday of this week.
Friday, there's gonna be the submission online button. So you can just close on this.
Friday online button. Submit your best papers, not the ones that were rejected by Hong Kong. Okay. Submit your.
That's the joke by you.
Submit your best papers.
I haven't rejected most of the papers.
I think yes, I'm done.
Okay. So our fourth speaker is Professor Ali Shojaie from University of Washington. He will give a remote talk.
Hi, everyone.
Hi, Ali.
Sorry I could not be there due to some family health issues. I want to thank the organizers for putting this really interesting workshop together and for inviting me as well. I also want to thank them especially for putting me last on this particular session, because everything that was talked about before I probably needed in what I'm going to talk about today. So I'm going to talk about non-stationary brain imaging data, and in particular, sort of what we learn from changes in brain. And this is, you guys see my slides, right?
Yes.
Okay, perfect. So we have time series data that capture activities of brain regions at different scales, both temporal scales and spatial scales. And these are just some of the examples that we've seen throughout the past couple of days as well. At lowest level, we have spike train data capturing individual activities of individual neurons. We have mu ECOG arrays. I'll talk more about these in a little bit. fMRI data has been talked about quite a bit. EEG data that sort of a lot of people are familiar with already. And we've done work on all of these, and today, in particular, I'm going to talk about mute ECOG arrays and fMRI data. So I decided, after seeing Jen's interesting questions, I decided that's what I will mostly talk about two applications, and I tried to answer some of the questions that Jen was asking in the context of these two applications. And then hopefully at the end, I'll talk about some efforts that sort of we've been taking in order to address some of these questions. So the first application I'm going to talk about is analysis of fMRI data. And this is something that I've become much more involved in over the past 3 years since I've taken over the data management and statistics core of the UW Alzheimer's Disease Research Center. And now I'm much more directly involved in some of the basic questions from data collection and data processing to analysis, in collaboration, of course, with a large group of people really that are at UW ADRC. But sort of in particular, what I'm going to talk about today is approaches that sort of are commonly used for analyzing fMRI data in order to understand AD pathology and sort of maybe develop biomarkers for AD. So this is a typical pipeline that sort of we process, for example, resting state fMRI data, we map it to some atlas. In this particular example that I'm going to show you, we look at sort of it really, this is a proof of concept study. We look at the default mode network, the DMN network. And so we perform some analysis of the functional connectivity, and then using differences in functional connectivity, we try to either understand the disease pathology and or develop some biomarkers, for example, for Alzheimer's disease subjects versus those who are cognitively normal, or MCI. And commonly, the assumption here is that sort of the time series are stationary. And yesterday, John talked about this briefly, and Sumanta talked about it a little bit more today, that there are approaches for considering non-stationarity, and there people have used these approaches before to sort of basically model non-stationarity in these time series. And in my view, sort of assuming stationarity a lot of times is at best suspect. I mean, when someone's sitting and sort of lying down in a scan for 5 minutes or so, assuming that sort of their state of mind is stationary, that's probably a big assumption. And so what I'm going to talk about today are sort of ideas around sort of what happens when the data is non-stationary. So in this sort of pipeline, when we look at this type of pipeline, when we try to see whether the, for example, correlations between these nodes in the default mode network are constant over time or not, we looked at, I'll talk more about this data. So this is data from UW ADRC. This is resting state fMRI data, and we look at correlations between 4 different pairs of nodes. And I've put the pairs here, it's PCC with DPC left, et cetera, et cetera. And we see that so the correlations are changing over time. Some of them are changing quite drastically over time. So this is the way that we got these estimates, is the common approach of basically taking a sliding window approach. That's so you calculate correlations over sliding window and you recalculate them. And so you see that the correlations are changing over time. So just by looking across different subjects at these correlations, we see that the correlations change, and sort of they change more for some of the node pairs than the others. However, this sliding window approach is not really very appealing. One, choosing that window length is difficult always, but also sort of, especially when we consider so multiple subjects, the analysis with these like sliding window approaches becomes more complicated. So instead of using this approach, what we have done, again, this is a very sort of proof of concept study. What we've done is that sort of we've used data, we've used this approach, the GARCH modeling approach, which is basically models conditional heteroscedasticity in time series, and sort of it originally developed was originally developed for analyzing volatility data, but it's been also used in neuroscience settings, including work 10 years ago by Martin that sort of they considered this DCC Gauch model, which basically models time-varying covariances. That it's a really simple and efficient model for capturing how correlation structures at time t depend on the past correlation structures. So we could model the changes in the dynamics of the correlations. And this particular model is appealing because it has two parameters that basically tau1 and tau2, that the first one, tau1, determines how the correlations vary as response to new stimuli. And tau2 determine how the degree of dependence on the past correlations, basically, how sticky the correlation is, as it's called. So what we did was that we went and we used this model and applied it to the default mode network. Default mode networks is small network, so we could you don't have to worry about high dimensionality, etc. And it's a well-characterized network that is known to be associated with Alzheimer's disease. And just to tell you a little bit more about data, this is resting fMRI data from 87 subjects aged between 54 and 91. And the reason that we chose these subjects in particular is that sort of for these subjects, we have CSF A beta assays as well. So for these subjects, we could determine whether they're amyloid beta positive or negative. This is done a little bit, I mean, this is what's done in the field based on a cutoff of A beta 42 over A beta 40. And so that cutoff value is 0.11. I still don't understand exactly why they take that cutoff value. And we don't need to really analyze this as categorical A beta positive or A beta negative, but sort of clinicians, and this is in the spirit of collaboration, this is what they were interested in, to see whether we could identify imaging-based biomarkers for amyloid beta deposition. And this is important because the new developments in therapies that we have for treating Alzheimer's disease, they're only effective in the pre-symptomatic subjects. So there's debate whether they're effective or not in general. There's been a long debate over this. But if there is any evidence for their effectiveness, it's really in people who don't yet demonstrate any cognitive decline. So the question is, can we identify biomarkers early on based on the presence of amyloid beta, maybe before they show any signs of cognitive decline, they're at risk of basically dementia. And CSF is a good biomarker. It's a very sort of specific biomarker, and we had conversations about this yesterday as well. But CSF requires a lumbar puncture, and it's because of that, it's an invasive procedure. So patients are not always willing to go under this procedure to collect the data. So it would be interesting to develop imaging-based biomarkers that are readily available and could be used in clinic more easily to identify people who might be at risk of Alzheimer's. So we did this, and of course, we could see already that there are differences between the networks of A beta negative patients and A beta positive patients. Hernando talked about heterogeneity in these networks. This is actually a model that sort of we did based on mixed models. I'm happy to talk offline. You're absolutely right. It's difficult. It might not always work, but we could actually get it to work in some cases. This is one of those cases that we were able to develop a model that sort of characterizes not only the heterogeneity across subjects, but also we could do testing formally to identify sort of what are the edges at the population level are different. So these are the edges at the population level are different between A beta negative and A beta positive subjects. And we see, and this is consistent with what people have found out in the literature before, that sort of there are connections are lost within the DMN network in A beta positive subjects. Now, what was then interesting was what we found based on looking at these two parameters, tau1 and tau2. So we looked at the values of tau1 and tau2, and this is a small, relatively small study with only 87 subjects. And we see that between the two groups, we have marginally significant difference between the tau1 parameter for A beta positive patients versus A beta negative patients. So tau1 is smaller for A beta positive patients. Similarly, tau2 is larger for A beta positive patients. We don't have really significance at this for this parameter, but these two parameters are related, so they're not completely unrelated. And so what we did was that we then took it a step forward, and this is, I think, the first time, as far as I know, that this has been done. That so we just use these two parameters as a really simple biomarker. For each individual, we have this pair of tau1 and tau2. We said, can we predict whether they're going to be A beta positive or A beta negative based on these two parameters? And sort of we developed a simple model. And sort of what we see is that opposed to, in contrast to sort of the stationary network modeling that sort of basically gives an AUC that is almost like random guessing, we gain something by modeling through this dynamic correlation. And just using the tau1 and tau2, these really simple summaries, we gain quite a bit over sort of just random guessing. And this is a difficult problem because, again, so these are not patients who have developed Alzheimer's disease yet. These are pre-symptomatic sort of individuals that sort of, some of them are A beta positive, some of them are A beta negative. So there's some hope that sort of ideas along these lines might be, we might be able to use them to develop imaging-based biomarkers for detecting Alzheimer's at early stages and sort of guiding therapy. And so what we've been thinking about lately is to sort of basically change that analysis pipeline to now think about sort of dynamic functional connectivity analysis, because in a lot of existing approaches really treat non-stationarity as sort of a nuisance. They try to somehow model it correctly, which is better than sort of ignoring it completely. But what we're finding, at least based on this proof of concept study, and there are other studies in the field as well, is that the non-stationarity actually might be informative. In particular, what we found in this study is that individuals who are A beta positive, their correlations are stickier. They change less over sort of that exam period compared to individuals that are A beta negative. So what we're now working on is to extend these approaches to sort of develop a flexible high-dimensional gauge model in order to develop really hopefully better biomarkers for changes during the AD pathology. So that's one application. I don't know how much more time do I have, but I'm going to also tell you another application that is related to what. Oh wow! Okay, I'm taking my time then. So I'll talk briefly about sort of this one. This one's a animal study, and this is the case where we have interventions in the brain and through optogenetics. So this is in monkeys, and these monkeys are trained to do a particular task, and there's a new ECOG array that sort of is implanted on the brain. This is the actual image, and there are laser beams that sort of activate neuronal spike in those regions. So now we could actually do intervention while measuring the activities of the brain in this about sort of 100 locations. So we get functional connectivity estimates in this case, and so what we are interested in understanding is that sort of how functional connectivity changes as a result of a stimulation. This is very much related to sort of what Hernando was talking about. And so what we've done here, I'll have to go a little bit more quickly, is that sort of what we realize is that unlike the previous sort of this is in collaboration with colleagues at UTHealth, what they were doing before this lab, they were using the experimental protocols to see if they could predict how the brain connectivity is going to change. And what we realized was that that's not enough. We need to take into account the current state of connectivity in order to predict how the connectivity is going to change. In other words, this is very subject specific. Each subject's connectivity changes based on their current status and not just based on experimental protocols. And so I will skip over sort of the modeling details of how we got to this point, but sort of we use like flexible models that sort of, in addition to sort of network features, and this is, Jarek talked about this. So yesterday we used higher order sort of network features and changes in those in order to be able to model changes in connectivity. And based on this, we basically get much better R squared. Still sort of pretty dismal if you consider these are squared values, but sort of much better than sort of just based using the experimental protocols alone. And these are test R squareds. So here's another example that sort of really what we're interested in, yes,
To wrap
is
up,
the
I'm
change in
sorry.
connectivity. Yeah, no, I'll wrap it up. And sort of the goal here is causal. So I just want to mention this. Eventually, what we want to build in this, and this is a cartoon, we want to build it and control it to be able to really control how the monkey's behavior changes, but that's a long-term project. So I'll wrap it up and I won't talk about sort of these other stuff, but I'll just say that in response to Jen's question, I think there's actually a lot of opportunities. I'm a little bit of an optimist. There's a lot of opportunities in modeling time series data in neuroscience and pay attention to non-stationarity. There's opportunities for new discoveries and new biomarkers, etc. There are lots of challenges that some of them have been discussed yesterday: data harmonization, data integration. I think validation is something that I've always struggled with. So I know that this came up in some of our conversations today, but I think validation is something that's difficult. I'll stop there. Thank you guys for having me talk here.
Any questions? Yes, Hongzhu?
When you run the models, grammar models is on the dynamic functional connectivity across the subjects, right? Your input data.
Hongzhu, I'm not sure if I heard your question completely. So you're asking if we could run dynamic functional connectivity across subjects?
No, the model you feed to the data, that response is the dynamic functional connectivity, right? The model.
Yes.
Okay. But
yes.
The only one issue is because I think the subject they may have the time drift across the subject. That's the could influence the result.
Yeah, absolutely. So that's actually no, you're absolutely right. I was hoping that I would have enough time to talk about this, but I think that's actually a really important issue about sort of I've added here in terms of challenges, heterogeneity and or generalizability. I think there are differences not only in sort of time, there are many differences in terms of how we could generalize from a single individual to multiple individuals and sort of across multiple studies. I think you're absolutely right.
So just to confirm, so the model is applied to a single subject fMRI data? So apply to it.
So the Gauch model is applied to each subject separately.
Or each subject. Okay.
Yeah, the Gauch model is applied to each subject separately. So we get estimates tau 1 and tau 2. We have a time series for each subject. We get an estimate for tau 1 and tau 2 for each subject separately.
Okay. Yes.
Yeah, so firstly, for really cool talks, thank you. Rossi, I have a question. Like, where does the information for delta come from? Like, where?
Okay. Yeah. So in our model, it really comes from if you're looking at a single subject, we really have a single level. You cannot have enough information to identify data that has been proven by the literature. So what we have done is really to leveraging the multi-subjects, multiple sessions, and using that kind of multi-level data. So we have, to some extent, we are leveraging the heterogeneities across subjects to help identify what to attribute based on based on the mixed type models, to attribute like how much of the variation is due to the subject variation, how much of the variation is due to the between the mediator and outcome variation. So that helps us identify the components of the magnitude of the delta. Yeah, I hope that makes sense. Because we have sort of like repeated, more like using the multiple subjects, we have more measures of the same delta across subjects and across sessions. So if you have a single subject, then essentially reduces to the classical literature has shown is like you have only one measure of the delta. Of course, you cannot estimate the covariance using a single observation. But because we have multiple subjects, multiple sessions, based on the hierarchical models, now we have more observations to identify the covariance structures. Yeah.
Does that assume that the delta is the same across all?
Yes, with certain limitations, we does have to require some kind of repeated measures, observations, so depending on the experiments are set up. So you have to, either assume they are the same for the session or some kind of constant. So in order to identify it, otherwise it won't be possible. If you assume the delta is different across subject, across sessions, across trials, and then we reduce it to the classical situation that people have shown it's impossible. Thank you.
Yes.
My question is for Shilo. I may have misunderstood the delta. Is that your assumption, or is that something you can confirm based on your data?
Okay, so the delta is really a mathematical, abstraction of the or some kind of summary statistics for the unmeasured confounding. We're trying to use that to quantify and capture the magnitude of unmeasured confounding and between two brain regions. So we know like there are many brain regions connected to those. So yeah, it's a simplified model and it may not capture all the dynamics of the magnitude or varying unmeasured confounding. So
in that case, is it fair to say that you are proving Julia Pearl's argument? You do need some assumption, some theory.
Yes, I never
derive causality based on data alone. So you do need some theoretical assumption.
I'm not disputing that. I'm just saying that within the neuroimaging community, especially related to my research in fMRI, there are some more reasonable assumptions rather than just saying the two brain regions, they don't connect to anything else, so the delta is 0. So what we are trying to say is, if you, I know like you work on mixed effect models, if you believe like those models, do capture a lot of the variations, and if you can using those model type of assumptions to replace, in my opinion, a little bit more subjective assumptions, say those two brain regions are not connected to anything else. So I feel like that's a little bit better. Yeah.
Your model is built on the mediation effects is built on the both signal level.
Yes.
It's reasonable to say the mediation effect happens at the neural activity level, not at the both level. So in that sense, if your activity is mediated by multiple factors such as neurovasculature, physiologic components, even head motion. Right? So when you consider that, then
I mean I don't dispute with that. So we are trying to be a bit careful with that as well, and using fMRI, of course. I think it's always challenging to claim like causality, like you said, those like both effects. So for some of the models, and we try to like do the deconvolutions and try and do the best. And but I guess there is still a lot of room trying to improve our work. That's why I always think like validation, how to. I feel like this is really a baby step, and also currently some other projects we also think integrating of other modalities and probably more closer to measure of those neuronal signals, and so hopefully bring multiple modalities together, integrating together will help us better resolve those issues. Yeah, thank you.
I have a quick follow-up question. So how much variability you can expand in your real data analysis from the mediator to outcome and exposure to mediator?
I think to that question, I have to say is because we have multiple variation components, and the one that we mostly care a lot about, because we understand there are a lot of subject variations and setting of variations. So a lot of the variations we care about is really about sort of like the ratio, the percentage mediated. So in that sense, what we have shown is like the one brain region really have contributed a lot more than, I think more than 50% of the mediated. And so I think to your original question, it's the fMRI signal is really noisy, so it only helps a little bit better fits in terms of model fitting or decrease the variation, but I think there is still long way to go.
50% is huge.
In terms of percentage mediated, so.
Okay, so yeah, I guess so. Actually, I have a question for Hernando. This maybe leave for the panel is like since Hernando is the editor for the new journal. I'm just curious, what's a different focus of that journal so that it can compete with AOS and the JASA application. Yeah.
Out here.
I don't know if I'm the best person to explain this, but maybe you can also hear from both Martin and Michele who are here. The whole point of this is to have a sounding board of ideas to be listened to by fellow.
I tell you the reason.
How do you know the reason?
Because I can only accept about 5. You can accept more than that.
Well, we want papers to be high quality. We want papers to be high impact. We want the papers to encompass more than just neuroimaging. In fact, our first paper is going to be the solar imaging. So we want all kinds of imaging. We're going to have experts. We'll try to make the journal to be somehow a little bit more interactive. You know, the details are being worked out. We'd like the journal to be responsive to ideas. We don't want to be the nitty-gritty type of a journal. We want big ideas. So ones that can really move the field. We want big ideas. Yeah.
I think the key difference is because JASA every year can only publish about 50 papers in JASA cover all the topics. And that's the reason I tell you 10%. That's your anticipation from the imaging side. Okay. Then most of the other papers, the machine LS can publish around 160 something. And you think about this, all cover all the topics. That means about 10%. Okay. That means about every year, about 16 papers publishing LS on neuroimaging related. That's most others that give to you. Thank you.
I guess we can conclude this session. We'll move on to the panel.